Welcome back, my students, to chapter 6. The second topic is protein. Proteins are polymers from 50 to 5,000 amino acids joined by peptide bonds. The peptide bond is the bond between the carboxyl group COH of one amino acid and the amino group NH2 of another amino acid. So this is the carboxyl group C double bond OH, and this is the amino group NH2. Remove OH from the carboxyl group with H from the amino group as H2O. Now join the rest of these two compounds, C double bond O with NH, so you will get C double bond O NH. This one we call it peptide bond. So peptide bond is the bond between the carboxyl group of one amino acid with amino group of another amino acid. They condense by removing H2O and give us the peptide bond. So proteins contain many molecules of amino acids from 50 to 5,000 joined by this bond, what you call it, peptide bond. Now condensation of amino acid gives what? Yes, gives proteins. What is the building unit of protein? The building block or the building unit of protein is alpha amino acid. Here we have an example. If we have glycine, as we studied in chapter 5, and alanine condensed to form glycylalanine. This type we call it dipeptide. So now this is the glycine and this is the alanine. From the glycine remove OH and from the alanine remove H. Then they are removed as H2O. Join the rest of the compound. From here we have C double bond O joined with NH. So this is C double bond O NH. So this is what we call it? Yes, peptide bond. Now I have two amino acids are joined with each other. So the type of the compound, what you call it? Dipeptide. Di, it means you have two amino acids. And this di related to how many amino acids? Not, to the bond, because I have only one bond, one peptide bond. But the family of this compound is called dipeptide. So di related to how many amino acid? So I have two amino acids, that's why we call it dipeptide, but we have only one peptide bond. The name for this compound, because I remove OH from the glycine, that's okay, I remove I-N-E and replace it by YL in the name. So from glycine changes to glycyl. And the one that you remove H, it is the same name as what? As alanine. So the compound, the name is glycylalanine. But the type of this compound is dipeptide. Why? Because it contains two amino acids. And how many peptide bond? We have only one peptide bond. Okay, now if you have two amino acids, so condense forming what? Yes, dipeptide. Is it protein? Up to now, no. If we have three amino acids, yes, give us what? Tripeptide. If you have many amino acids, from 50 to 5,000, that's why we call it, in this case, we call it proteins. So by repeating this process, we can get a polypeptide. Poly means many peptides bond, what we call it polymer, and this polymer, it is what? It is protein. So protein is polymer of polypeptide. Yes, that's true. What is the structure of proteins? Protein is an organic compound, contains carbon, hydrogen, nitrogen, and oxygen, and sometimes sulfur and phosphorus in small amounts. 16% of the weight of protein is nitrogen. So this is a very important point. The compound that 16% of its weight is nitrogen, what you call it? Yes, it is protein. Now, how many types of alpha amino acid that we studied in chapter 5? Do you remember it? Yes. There are 20 types of alpha amino acid required for building proteins. They can be classified into two classes. What are they? Yes, they are essential amino acids, and we have 8. These essential amino acids can't be synthesized by the body, so they are taken from the food containing proteins. They are very important for the growth of the body. They are found in animal proteins in high amount. In this case, why they are essential? Because your body cannot be synthesized. They cannot be synthesized by what? By the body. So your body cannot synthesize what? Synthesize them. That's why they are essential. You have to take them from the food. That's why they are essential, to take them from the food, because they are very important for your body. So what is the best source of them? The best source of them is animal proteins. So animal proteins are better than plant proteins. Why? Because they give, or they contain, high percentage of essential amino acids. Here we have the type of the alpha amino acids, essential alpha amino acids. We have leucine, phenylalanine, valine, threonine, methionine, lysine, isoleucine, and tryptophan. So these are the 8 essential alpha amino acids. Here we have the sum structure of some of them. This is for the phenylalanine, for isoleucine, for tryptophan, for leucine, for lysine. And as we see here, all of them, they have the same, yes, part in H2CH, double bond OH. This is alpha because amino group at alpha carbon, the carbon that adjacent the carboxyl group C double bond OH. And here it is the same, here the same. All of them, they have the same, yes, CH and H2COOH. That's why this is, you call them what? Alpha amino, because amino group at alpha carbon, the carbon that besides C double bond OH, or the carboxyl group. The second type are non-essential amino acids, and we have 12. They can be synthesized by the body. That's why they are what? They are non-essential. Okay. They build the protein of tissues of the body. What is the importance of proteins? Proteins are the most important constituents of human food. Yes, they build the living tissues and regulate the metabolic activities in tissues. Proteins are used in building skin, hairs, nails, and the muscles. Yes. How many sources of protein do we have? we have two sources, natural sources, animal proteins. They are found in meat, eggs, and milk. The second source is plant proteins. They are found in the legumes such as beans, now we have the experiment for detection proteins in your urine. The proteins in urine is called albumin. So we have to boil your urine. If the color of urine changes to milky or cloudy, it means turbidity is seen. Maybe you have albumin or phosphate. Then add 2% acetic acid. If the turbidity doesn't change, that means it is due to the albumin in what? In proteins. If the turbidity disappears, that means it is due to phosphate and you don't have albumin in what? In your urine. Again, how can you know you have protein or albumin in what? In your urine? Just boil your urine. If you see the cloudy solution, so add acetic acid. If the cloudy solution doesn't change, that means you have albumin or protein in your urine. The second experiment is detection of proteins in egg whites, the albumin in eggs. In this case, you have to add nitric acid, HNO3, to the albumin of eggs. It is the egg whites. If you see the white precipitate, that means you have albumin, and this one detects albumin, which changes to orange by adding ammonium hydroxide, NH4OH. So maybe the question is, nitric acid and ammonium hydroxide are used to distinguish or to detect, yes, carbohydrates or proteins or vitamins or lipids. What you choose? Yes, you have to choose, yes, proteins. By the way, HNO3 and NH4OH can be added to your urine. You will get the same result, white precipitate, then changes to orange by adding NH4OH, and also for the milk, because all of them contain proteins. Thank you for listening.